right, everybody, welcome today to lab number 4. These are enzymes. We've already talked a little bit about what enzymes were in our last lab. Now we're going to study them in a bit more detail. So as you recall, enzymes are proteins. So that means that what we've already talked about, proteins, already still applies. They'd be stained with Biuret solution. You can, they're made up of amino acids, but these are proteins that catalyze chemical reactions. To catalyze means they speed chemical reactions up. But the cool thing is that they can speed them up and make them happen without heating the cell up. A lot of times, the reactions that a cell does, if they were to occur in nature, would actually generate a lot of heat. And enzymes are able to cause the reactions to happen quickly, but without releasing all the heat. Now, when we look at enzyme reactions, we're going to talk a bit about what we call substrates and products. So the substrate is the chemical that the enzyme binds. It's what it's going to work on. And the product is what is the result of the chemical reaction. So let's take a look here at this little thing here. You can see what we have going on is, let's do it over again. Here are these two green things. We're going to fit into the active site right here, and then they get broken apart. They're cleaved. So what happens is that the way that enzymes work is that they have an active site. This is the active site right here, and it's the part of the enzyme that binds the substrate. The substrate was that green molecule right here. And the active sites are extremely specific. They are specific to that substrate and only that substrate. So for instance, if you have an active site for sucrose, that is a disaccharide, it won't fit lactose, which is another disaccharide. So they are going to be very specific to the chemical that is the substrate. That's what we call substrate specificity. And these work together sort of like a lock and a key. So if you think of it this way, in terms of a lock and a key, so here, I have, hope you can't see that very well. Here is the key to my office. The key to my office is specific to my office, but it will not open the door of my house. I need a different key for that. This is sort of the way that enzymes work. They're going to be specific to the substrates that they work on, and that's the substrate specificity. Okay, now something that can happen is also what we call denaturation. So this is a permanent structural change to an enzyme that makes it inactive. So if we look here, let's see if I can get this to run again. This bump right here causes that substrate to not be able to fit in. Okay, here we go. It fits in and breaks apart. But when the enzyme denatures, see there's a bump here, there's a change in the shape, and this product can't fit into the site anymore. So denaturation is a permanent structural change to an enzyme that makes it inactive. The important thing is that it's permanent. Enzymes are very complex. They can be turned on and off in the presence or absence of certain things, but those are reversible. Denaturation is permanent. And things that can cause denaturation include high temperatures. If you heat a really good example of this is an egg. The egg white is mostly protein. And in the raw egg, it's clear. But if you heat it up, it turns white and opaque. And that's a permanent change. Once you've denatured those proteins, you can't put them back into the clear form again. Other things that can cause this are a change in pH. So if it's too acidic or too basic, it can actually destroy the protein. So with that, let's go ahead and walk through your activities today. Let's start with exercise number 1. So for exercise number 1, we're going to test the effective enzymes on starch. And we're going to be using the iodine potassium iodide test, that ICI test. If you don't remember that from last lab, go back and review iodine potassium iodide and Benedict's solution, because we're going to use both of those in today's lab. So what we did is, here we go. We have 3 test tubes here. each of them has starch and each of them has iodine, potassium iodide. So that's why you can see they've all turned black. So this tube is starch and iodine, potassium iodide. This is starch plus saliva and the IKI. And here we have starch plus over here, this is rennet or renin. This is another enzyme. And so what we did is we let this sit for 10 minutes. And after 10 minutes, well, here's what it looks like. So now, based on what you know about iodine, potassium iodide, you need to go and figure out what is happening. So go to open up the assignment PowerPoint, and there will be a table. Fill in your answers to the table. And then there are going to be a series of questions on the next slide. You're going to write in your answers to that. When you're done with that, you have finished your first exercise. So in essence, what you're really looking for is, why did this one change color and why did those two not? All right. Once you're done with that, we're going to go on to exercise 2. This is the effect of enzymes on starch, but again, we're going to change the test. Now we're going to use a Benedict's test. So again, if you don't remember Benedict's test, you'll want to go back and review that from our previous lab. So here are the results. So what I want you to do is to interpret these results based on that. Oh, excuse me. I'm going to pause you for a second. I've forgotten to add some important information in here. All right, sorry about that. I added in the labels here so it's so easier to figure out what we're looking at here. So this tube here on the left, this contained starch alone plus the Benedict solution. This tube in the middle had starch and saliva. And this tube to the right had starch and renin. Again, renin is another protein. It's another enzyme, and its function is it has you can read about the function in the lab itself. Okay, so what I want you to do is interpret these results. And then once you've done that, you're going to fill in your answers in the table on the assignment PowerPoint, and then you'll answer a couple of questions. And once you're done with that, you are free to move on to our third experiment. All right, this is experiment number 3, or exercise 3. This is going to be the effect of enzyme concentration on catalase activity. So there's some important things we need to know about before we get started on this one. The first is we need to know what hydrogen peroxide is. It's H2O2 is the chemical formula, and hydrogen peroxide occurs as a side effect of many cellular processes. And so this happens, it just happens all the time. The problem is that hydrogen peroxide is actually toxic to cells. So if the cell has a buildup of too much hydrogen peroxide, it will actually kill the cell. And this, of course, is undesirable. And so cells have an enzyme that will break down hydrogen peroxide. This enzyme is called catalase. So what catalase does, it takes hydrogen peroxide and it breaks it apart into water and oxygen gas. Now this enzyme catalase occurs naturally in yeast. It occurs in plant cells, and it occurs in animal liver cells. So your cells actually, your liver is actually producing catalase. That's one of its jobs. And that catalase then will be used to break down harmful hydrogen peroxide. Today, in today's experiment, we're going to use yeast as our source for catalase. So we can do yeast. Another common one is potatoes are very popular. And if you really want to, you can actually do similar things with liver. But getting fresh liver is a bit more difficult. Okay, so what we're going to do is we're going to use the catalase from the yeast, and we're going to run an experiment. And so here we go. You can see there are several things that you'll want to look at for this experiment. Right here, we have some tubes. You can see this tube here to the left is a lot more clear than the tube here to the far right, which is much more cloudy. What we have here are different concentrations of yeast. This is a 0.5 concentration, a 1% concentration, a 1.5, a 2%, and a 3% concentration. So we have different yeast concentrations. And then here we have little paper discs. These are from filter paper. And so what I did is I hole punched some filter paper. And what we're going to do is we're going to actually take these discs, filter paper discs, with a pair of forceps. We're going to dip it into the yeast solution. And then we're going to drop the disc into, here's a little beaker with 30 milliliters of hydrogen peroxide. And what's going to happen is that when you first put the disc in, it's going to sink to the bottom, but after some time, what will happen is that the little disc will float to the top. So what we're going to do is we will time how long that takes. So if you want to see what this actually looks like, here is a 40-second video that you can go to on YouTube, and you can watch somebody stick one of those discs with yeast into the hydrogen peroxide. You can watch it float. That's essentially what I did with all those concentrations. But then I actually took the data and I've made you a data sheet, and it's going to be called the catalase data sheet that's done in Excel. So what you're going to need to do is you're going to open that catalase data Excel sheet, and you're going to follow the instructions in the lab for making a graph. You're going to graph these results. You're going to take that graph, and you're going to then, for the last bit of your assignment, you're going to copy and paste that graph into the PowerPoint. And once you've done that and you've answered all the other questions, you have finished your enzymes lab, and we'll see you for lab number 5.